So there's lots and lots and lots of reasons for optimism. We've got an exponentially growing knowledge base. It's growing by leaps and bounds. We've got wonderful insights for the future. We've got a new technology base. We've got gene chips and all these other things. We have a vibrant, efficient, outstanding research community that's tops in the world in impact and in cost effectiveness. We have unlimited opportunities. And so the question, of course, is, what are we actually doing with all these golden assets? Stem cells in this context really just means any cell that can give rise to a whole lot of other cells of the same type. Think of it in the blood system. We have all kinds of cells. We have red blood cells, white blood cells, platelets, this, that. They all start from one cell type, sort of an Adam and an Eve kind of cell that gives rise to all of those other ones. That's the stem cell. That's what we're talking about. And in fact, the modern era of stem cell research started right here in Canada back in the 60s when Jim Till and Bun McCullough published the very first assay or test to actually prove that stem cells existed in the bone marrow. We still lead the world in this area. Two of the current gurus in this is Dr. John Dick in Toronto and Dr. Mick Badia in Hamilton. I can tell you there's not a university or institution anywhere in the world wouldn't love to capture either one of these guys, and they've chosen to stay in Canada. And I've got a couple of slides that I borrowed from John to give you an idea of what we're talking about. In the traditional model of thinking about tumors, we know that a tumor may be made up of lots of different kinds of cells. You can see they're in different colors and so on. And we've always believed that every one of those cells, if taken out of the tumor, could give rise to another tumor. Well, the stem cell model says, folks, that actually most of those cells may have the ability to divide rapidly, but most of them don't actually have the ability to recreate all of the tumor characteristics. And that in fact only a very tiny subset called these stem cells are the ones that have this potential built in. And this has a very significant therapeutic implication for exactly the reason I mentioned. You can wipe out the bulk tumor, but if you don't get the stem cells, you have the ability for that tumor to regrow. So it's not enough now to say you've identified the haystack, let's get rid of it. You have to now find the needle in the haystack and make sure you get rid of that too. And so a lot of the current research is in looking, how do we recognize that stem cell? How do we recognize the needle that looks different than all the rest? What characteristics does it have? How can we exploit that? How can we find it? How can we get rid of the stem cell? Let's stop concentrating on the bulk tumor and let's start concentrating on the stem cell, because that's the brains of the operation and that's what we have to get. And I can tell you Canada is leading that fight.